In the previous two web lectures, we explored just the anatomy of the cell. We went through kind of a whirlwind tour of the different parts that make up a cell. And in the next couple of sections, we're going to start looking more in depth at some of these components and look at the specifics of how they do the things they do. So we're going to begin with the plasma membrane and look at the structure and function of the plasma membrane. In this web lecture, I'm going to focus primarily on its structure and how that structure contributes to the main function of regulating traffic of materials into and out of the cell. In the next web lecture, I will focus more on the mechanisms of how materials either move or are moved into and out of the cell. So let's take a look at the membrane structure first. So to review, the plasma membrane or cell membrane separates life from non-life. This is the boundary of that smallest unit of what can be considered alive. And it separates the cell's interior from the external environment, again, as we've said over and over, giving it the ability to maintain conditions within the cell within the narrow range necessary for its life processes. So membranes are going to function to keep damaging materials out of the cell, allow entry of materials needed by the cell, and they're also going to play a role in facilitating some chemical reactions, and we'll talk a little bit more about that in this web lecture, and a lot more about that as we get to things like photosynthesis and metabolism, where these membrane-bound proteins are really going to come into play. So the defining feature of the cell membrane structure is the fluid mosaic model of the lipid and protein components of the cell membrane. So the plasma membrane exhibits selective permeability. This is kind of the defining feature of its function. That means it allows some substances to cross it more easily than others. As we know from our segment on lipids, phospholipids are amphipathic molecules. That means that they contain a hydrophobic and hydrophilic region. And this is very important to the structure of this membrane as a phospholipid bilayer. The hydrophobic tails of the phospholipids are sheltered inside the membrane, while the hydrophilic heads are exposed to water on either side. Because all of these heads are going to tend to try to be in contact with water, it's going to form a single layer of hydrophilic heads on the external surface, a single layer of hydrophilic heads on the internal surface of this membrane, being exposed to water on either side. So in terms of the components of the plasma membrane, phospholipids are going to be the most abundant lipid in the plasma membrane. There will be some other lipids incorporated, as we'll see later. And then there are also going to be different proteins embedded in this phospholipid bilayer, and they're also going to play a role in controlling the transport of different materials into and out of the cell. So before we go too much into the structure and function relationships, let's think a little bit about how do we know what we know? Always an important question. How do we study phospholipid bilayers? So we mentioned when we first started talking about phospholipids way back in the chemistry section of the course, when amphipathic lipids are placed in water, they're going to spontaneously assemble into certain forms. So the hydrophilic heads are always going to interact with water. The hydrophobic tails are always going to interact with each other away from the water, and this is going to result in the formation of structures either in the form of micelles, which is basically a little bubble that just has a single layer of phospholipids with all the tails pointing in to the center of this bubble and all of those hydrophilic heads pointing out. This is important in the way detergents work to break up lipids into little particles that can be washed away. Or they're going to form lipid bilayers. Again, the hydrophilic heads interact with the water on both sides. The tails are safely sandwiched in between those two layers of heads. So we can assemble these phospholipid bilayers in a couple of different ways. So if you just drop these phospholipids into water, they're going to tend to form little vesicles. They're going to self-assemble into a little droplet with water on the inside and water on the outside. And these artificial membrane-bound vesicles we call liposomes. So these little small bubble-like structures floating in the water. And here's an image, electron micrograph image of these little artificial vesicles used to study phospholipid bilayers. But the other way we can structure these bilayers is in a plane attached to a hole in a glass or plastic wall. So what does this allow us to do? We can have two chambers in a container separated with this lipid bilayer, allowing passage if it's permeable to whatever you're adding to it. You can add a solute on one side and then study whether that solute ends up on the other side of the beaker, whether that phospholipid bilayer allowed passage of that solute or not. So we can use this to measure the permeability of lipid bilayers. So, and we can do several different things. We can either study the phospholipid bilayer by itself and examine the role of these phospholipids in and of themselves in regulating permeability, but we can also use different types of phospholipids to see the effects of, for example, using phospholipids with fatty acid chains of different length, with different numbers of double bonds in the fatty acid tails, but we can also insert proteins or other molecules into that membrane and add them in a very specific way to be able to study their effect on the permeability of that membrane. So first, let's examine the fluidity of membranes. So what does fluidity mean? So the phospholipids in a plasma membrane will kind of move laterally about within the bilayer. So as part of this motion of the lipids in the bilayer, they're mostly going to move around laterally, but very, very rarely one will flip and end up on the other bilayer, but that is a very, very rare occurrence. Why do they do this? Because they have some thermal energy that's causing random motion of molecules, and they're going to tend to move around in a more or less random way just due to having thermokinetic energy. So, because this is related to thermokinetic energy, it's going to depend quite a bit on temperature. At lower temperatures, the molecules in the bilayer are going to move more slowly. They've got less of that thermokinetic energy, and because of this decrease in movement, those hydrophobic tails are going to tend to pack together more tightly. So this is going to have an effect on both the fluidity and the permeability of the membrane. So as temperatures cool, membranes switch from a more fluid state to a more solid state. We observe this in cooking materials that we use that are lipid-based. We can melt butter and turn it into a liquid state by raising the temperature. And the temperature at which a membrane solidifies or stops being so fluid and moving around depends somewhat on the types of lipids that are in those fatty acid chains. So this is exactly the same as what we were talking about with saturated and unsaturated fats. So membranes rich in unsaturated fatty acids, so there are one or more double bonds in those fatty acid tails, are going to be more fluid than those that are rich in saturated fatty acids. They're going to pack less tightly and they're going to have more room to move around relative to each other. Whereas these saturated tails are going to be very tightly packed. It's going to be harder for them to move. So membranes have to be fluid to work properly. We're going to talk more about the proteins that are embedded in these, but they need to be in the right place at the right time, so they need to be able to move around within this bilayer. Typically, membranes are usually about the fluidity of salad oil, so you can think of that kind of viscosity when you think of these cell membranes. Remember, that's pretty fluid, but remember they've got this meshwork of cytoskeleton just underneath the membrane that helps to maintain the cell's shape. Another thing that's going to affect the fluidity of these membranes is the presence of cholesterol. So cholesterol is present only in animal cells, and the effect of this cholesterol in the cell membranes is going to depend on the temperature. So at warm temperatures, where normally this cell membrane would be becoming more fluid and move around more, cholesterol is going to restrain the movement of those phospholipids and make it more solid and less fluid. At cool temperatures, where normally these phospholipids would not be moving so much and would be starting to pack more closely and becoming more solid, it's going to maintain fluidity by preventing that tight packing and giving some space in between those phospholipid molecules. So we can say that cholesterol works as sort of a thermal buffer to maintain consistent fluid properties in the membrane in spite of fluctuations in temperature, and animal cells can have more or less cholesterol in those cell membranes depending on the need to moderate the effects of these temperature fluctuations in particular cells or in particular species. So in the fluid mosaic model, the membrane is a mosaic of protein molecules that are sort of bobbing around in this fluid bilayer of phospholipids. So these proteins are going to be able to move around. How do we know that they move around in the membrane? You saw this experiment in your book where a mouse cell was fused together with a human cell and left alone for a while. And when we go back and check these tagged proteins on the cells, tagged specific to the mouse or the human origin, we find that the proteins have distributed themselves across that fused cell. The mouse proteins didn't stay on the half of the cell that originated with the original mouse cell, and the human proteins didn't stay on the half of the cell that was of human origin. They floated around and mixed, so we know that these proteins move around within this bilayer. So it's a mosaic, but the proteins are not randomly distributed in the membrane. So just as if we think of a tile mosaic, these tiles are not randomly distributed. They are very carefully placed according to the image that the mosaic artist wanted to convey. Similarly, these proteins are distributed in the cell according to whatever function they need to carry out, being in the right place at the right time to perform its function. So let's think about the functions of these membrane proteins. What are they doing in there? So again, somewhat like a tile mosaic, a membrane is a collage of different proteins, often clustered in groups according to the function they need to carry out, and they are embedded in different ways in the fluid matrix of this lipid bilayer. So the main fabric of the membrane, the stuff that it's made of, is the phospholipids, but then the functional part of this, what's actually going to be doing the different jobs, is going to be these proteins that are in the membrane. So we have two different categories of proteins. There are peripheral proteins that are bound to the surface of the membrane, either the internal surface or the external surface. And then there are integral proteins that span the entire thickness of the membrane, and they're going to penetrate the hydrophobic core and have ends on both the external and the internal surface of the membrane. So integral proteins that span the membrane are called transmembrane proteins. So they're going to go all the way across the membrane. So this means that these proteins that are going to span the membrane have to be put together in a particular way. They're going to have the primary structure and secondary structure that allows them to penetrate this hydrophobic region within the interior of the cell membrane. So they're going to have to consist of one or more stretches of these nonpolar amino acids that are going to tend to stay within this nonpolar region within the cell membrane. So often these stretches of nonpolar amino acids are going to be coiled into alpha helices. And depending on the function of the protein, the structure of it can get very, very complicated. So first let's think about transport proteins in the membrane. So these are going to be proteins that have a channel in the center of them that allows very specific materials to flow through. So if you think about, particularly if this is a transport protein that transmits ions or other very, very strongly charged molecules, basically you have to have a protein structure that has hydrophilic region at both of the ends, the interior of the cell and the exterior of the cell where there's water. There have to be hydrophobic regions on the outside of this protein that are in contact with the fatty acid chains, the hydrophobic section of the membrane. But then on the interior of this channel, they have to be lined with hydrophilic amino acid residues that are going to allow charged particles to flow through. So again, the tertiary structure of these transport proteins are absolutely critical to their function and can become quite complicated. So in terms of transport proteins, we can have passive transport channels that are just kind of open holes in the protein that allows particular molecules to come through. We can have gated passive transport channels that don't use any energy but only allow materials through at particular times. We can have active transport channels that require ATP to move things in and out of the cell, all different kinds of transport proteins that can be in that cell membrane. They can perform enzymatic activity, so they can be embedded in the membrane in such a way that biochemical reactions that are in multiple steps, these can be lined up according to the biochemical pathway they're going to perform, and being in the membrane kind of helps keep all of those enzymes together and in the right order to perform all the different steps. Signal transduction is going to be a really important one when we get to, say, the endocrine system or the nervous system. So being able to attach some kind of signaling molecule and cause some kind of change in that protein that's going to transmit that signal down into the interior of the cell so that the cell can respond to it. Again, super important in terms of response to the environment. Cell-cell recognition we've talked about quite a bit with these glycoproteins, the proteins that have carbohydrate chains covalently bonded to them so other cells can recognize that they are perhaps part of the same multicellular body, or if these glycoproteins are on a bacterium that invades your cell, your body can recognize them as not belonging there and attack them. We talked in the last web lecture about these junctions between cells, so proteins that are embedded in the cell membrane are going to be responsible for this intercellular joining, things like tight junctions and gap junctions. And then also they're going to play a role in maintaining the position of the cell, anchoring different cell organelles into place within the cells. These proteins are going to have attachments either to elements in the extracellular matrix or parts of the cytoskeleton to maintain organization and structure of that cell. An interesting example of the importance of some of these cell surface proteins is given in your book, and I'm not going to go into a lot of detail, but a very minor change in the expression of one of these receptor proteins can make the difference between being susceptible to HIV infection or being pretty much immune to it, because the HIV virus is going to use these receptor proteins as a point of entry into the cell. And if it doesn't have that receptor protein, this co-receptor, in this case, the CCR5, it's not going to be able to bond and it's not going to be able to gain entry to the cell. So these can have huge implications in cell functioning and health of organisms. So these membranes, because they have these functional proteins within them, it's going to be important which is the inside and which is the outside of the membrane. If you have a protein turned around in the wrong way, it's not necessarily going to perform the function it's meant to perform. And so just as a reminder of this endomembrane system that's going to deliver these embedded proteins, these intrinsic proteins, out to the cell membrane, these proteins are going to be embedded in these little vesicles that come off of the endoplasmic reticulum and then come into the Golgi apparatus. They're going to move through the Golgi apparatus actually in the membrane, and then when that buds off, this protein is still going to be within the membrane. So remember that with these vesicles, because of the way they fuse and release their contents, the inside of the vesicle is going to end up being the outside of the cell membrane. So the proteins are going to be oriented in a particular way, and that's going to be determined by the arrangement that's made initially within the endoplasmic reticulum. So now let's think more about this idea of selective permeability. What is it about the structure of the membrane that brings that about, and what accounts for differences in permeability in different kinds of membranes? So all cells must exchange materials with its surrounding. So it needs to take in materials that it needs, either nutrients, in some cases oxygen, and it needs to get rid of metabolic wastes. And this process of exchanging materials with the surroundings are going to be controlled by the plasma membrane. So plasma membranes are selectively permeable. They're going to regulate the cell's molecular traffic. They're going to be responsible for maintaining those conditions within the cell that are going to allow life processes to happen. So if we first think about the resistance of the plasma membrane itself to materials moving through, the plasma membrane is going to have different levels of permeability to different kinds of molecules. If on the scale we have very high permeability, we can think of this as any individual molecule that comes and bumps into this membrane, the probability that that molecule is going to make it through. We can say that these small or nonpolar molecules are going to have a relatively high probability of getting through. Why? In the case of the nonpolar molecules, there's basically nothing holding them onto one side. They're not going to be attracted to all of these water molecules that are on the outside. As they move through, there's going to be nothing inhibiting them from moving all the way through and possibly coming out the other side. Remember, they're not particularly attracted or repelled by anything. Small molecules such as carbon dioxide are just going to have, due to their small size, some probability of just squeezing through. Less permeable to water, of course, that's also a very small molecule, which accounts for the fact that it has some permeability, but it's also going to be attracted to the water that is outside that cell membrane. It's going to be less likely to move through here. And things like glycerol are going to have even lower permeability. Large polar molecules that are polar but not charged, such as glucose and sucrose, are going to have even lower permeability, and fully charged ions are going to have very, very, very low probability of making it through that cell membrane. If that's kind of the baseline, what kinds of features of the cell membrane can cause the permeability to change? So many factors are going to influence the behavior of this membrane. So we mentioned the number of double bonds between the carbons and the phospholipids' hydrophobic tail. We mentioned this in terms of fluidity, that there's just going to be lots of gaps in between these phospholipids, a lot of space due to these kinks. And so this lower packing is going to make them more permeable. The more unsaturated those fatty acids are, the more permeable the membrane is going to be, as opposed to saturated fatty acids in those hydrophobic tails are going to be more resistant to passage of materials through the membrane because they're packed tightly together. The length of the tail is also going to be a factor. So the longer the distance the particle has to travel to get across the membrane, the less likely it is going to be to cross. So shorter tails, higher permeability; longer tails, lower permeability. And here we see the combined effect of saturation level and length of tail. Don't get confused. So if you have long tails and lots of unsaturated fats, that's going to affect the permeability also. Be less permeable as a result of the long tails, but a little bit more permeable as a result of the unsaturated fatty acids in there, less permeable than it would be if they were long tails and saturated fatty acids. And it turns out also that in animal cells, the presence of cholesterol reduces the membrane permeability. So we know this from experiments such as the one diagrammed here with artificial membranes, in this case in the form of these liposomes, that were constructed artificially in the lab to have different amounts of cholesterol embedded in those membranes. So either no cholesterol, a small amount of cholesterol, or a large amount of cholesterol. Some glycerol was added to the solution in which these liposomes were located, and the experimenters could measure how much of that glycerol ended up inside these little liposomes. So the results are here. When there is no cholesterol, there's very high permeability, and this was done at different temperatures, so the temperature is affecting the permeability, but at all temperatures, the absence of cholesterol led to higher permeability. A small amount of cholesterol gave it a little bit less permeability, and a larger amount of cholesterol gave it even lower permeability. So what's the reason for that? So adding cholesterol to the membranes increases the density of the hydrophobic section. Remember, we said that it's going to kind of increase the packing of those tails. It increases the membrane permeability by just increasing the density of this hydrophobic region and making it harder for materials to pass through, especially large and polar molecules. So the other thing that's going to greatly affect the permeability of membranes is the presence of transport proteins. So transport proteins allow passage of hydrophilic substances across the membrane. So I mentioned this briefly when we were looking at the different functions of these membrane proteins. So some transport proteins called channel proteins have a hydrophilic channel. So the core of that little pore in the middle of the protein is going to be hydrophilic. It's going to be polar. and certain molecules or ions can use it as a tunnel to get into and out of the cell. So for example, there are channel proteins called aquaporins that allow water to move in or out of the cell, and these are hugely important in many functions of plant cells. They're important in the function of mammalian kidneys. Use aquaporins to regulate the water content of urine. These are very important functionally. There are other transport proteins called carrier proteins that are going to bind to these molecules that are being transported into or out of the cell. And then they're going to change shape to shuttle them either from the outside to the inside or from the inside to the outside across the membrane. And these transport proteins are going to have very high specificity for particular molecules. So this is going to be the way that the cell can very tightly regulate the traffic of what's coming in and what's going out of the cell. So it can insert a lot of transport proteins at a time when it needs a particular nutrient, for example, to get it from the environment. It's going to synthesize a bunch of these transport proteins that are specific to that particular nutrient, send them out to the cell membrane, and so we can bring in a bunch of that nutrient. When we have enough of the nutrient, those transport proteins can be butted back off of the cell membrane and broken down and recycled. So in summary, if we think about the structure of the cell membrane, this is primarily characterized by the fluid mosaic model, in which the phospholipid bilayer is kind of the C, or this fluid component of it, with these proteins kind of floating around within this fluid structure. So the fluidity of the cell membrane is influenced by the number of double bonds in the fatty acid chains. Remember, it's going to move around more if the fatty acid chains are less tightly packed. So for saturated fatty acids, they're going to pack more tightly and move around less. It's going to be influenced by temperature, which is again going to influence the packing of those fatty acid chains. They're going to be more tightly packed at lower temperatures and be less fluid. They're going to be moving around more due to the kinetic energy at higher temperatures and be more fluid. And then we saw that the presence of cholesterol is going to influence the fluidity, and what that influence is going to be is going to depend on the temperature. So remember, the cholesterol is going to work as sort of a thermal buffer to maintain a constant fluidity in animal cells. So now if we think about the function of cell membrane, this is characterized by its main job, which is regulating the materials within the cell and the flow of materials into and out of the cell. So this is characterized by selective permeability. And the permeability is going to be influenced by, again, the number of double bonds in the fatty acid chain. More double bonds means that the fatty acids are less tightly packed. Things can move through more readily. The length of the fatty acid chains, so that will determine the thickness of the phospholipid bilayer. The thicker it is, the less likely things are to be able to move through. It's going to be influenced by the temperature. Remember, the temperature is going to influence the packing of those fatty acid tails. So lower temperatures, they're going to be more tightly packed, and the membrane will be less permeable or more resistant to movement across it. The presence of cholesterol. More cholesterol is going to make the membrane less permeable by allowing tighter packing in those hydrophobic tails, and then also the presence of transport proteins. So now that we know the ways in which the membrane itself is going to either allow or resist movement across it, in the next web lecture we're going to talk about what makes these molecules move in and out. What are the different forces that are going to be responsible for either the molecule moving in or out of the cell or being moved into or out of the cell. So stay tuned for that.